Good morning, everyone. So, Dr. Shaima here, again back to you all with the blood-brain barrier. The cerebrospinal fluid and the blood-brain barrier normally is done as one class, but I just wanted to, make a split between, just make it into two half-an-hour classes. So, when you look at blood-brain barrier, a scientist called as Paul Ehrlich, he had actually injected a dye which was called tryptophan blue into a living animal, and he found that it stained all the tissues except the brain. So this finding made him suggest that or made him believe that there is a barrier which prevented the entry of tryptophan blue into the brain tissue, and this barrier is what you call as the blood-brain barrier. So you have a picture there. Okay, now let's go to this. Now the blood-brain barrier is present between the cerebral capillary endothelial cells, and the blood CSF barrier is present between the epithelial cells in the choroid plexus. So need to be a little thorough there. The blood-brain barrier is present between the cerebral capillary and the endothelial cells. Okay, that's one category, the blood-brain barrier. Now you have one more, the blood CSF barrier. So what is that? That the blood CSF barrier is present between the epithelial cells in the choroid plexus. So there are basically two things which you all are learning, the blood-brain barrier and the blood CSF barrier. The tight junctions between the capillary endothelial cells and the choroid epithelial cells in the brain are acting as barriers and are similar functionally. Now these are formed during childhood. At the same time, the food processes of the astrocytes develop around the capillaries, and this process basically reinforces the barrier. So the term refers to a slow penetration or lack of penetration of a number of substances from the blood to the brain and to the CSF. So to go to the next slide, you can see the blood-brain barrier here. So one second, let me just go to the choroid plexus, which forms the blood-brain CSF barrier, and this is a pictorial representation of the blood-brain barrier. So let's just have a look at the features of the blood-brain barrier. Now the blood-brain barrier, if you see, is present between the blood and the interstitial fluid of the brain. The tight junctions between the capillary endothelial cells is supported by the astrocytes, which acts as a blood-brain barrier. Now the expanded food processes of the astrocyte incompletely it envelops the capillaries. I think that you should be seeing in this picture. So just have a look at that picture, the picture on top. So there you can see that it's a barrier which is present between the blood and the interstitial fluid of the brain. Now the tight junctions which are present in between the endothelial cells are supported by the astrocytes. Can you see that in the picture? The astrocytes are the supporting cells there, which are actually guarding the tight junctions between the endothelial cells, and it is the astrocytes which are acting as the blood-brain barrier. Now the expanded food processes of the astrocytes, can you see? They are actually incompletely enveloping the capillaries. They don't envelop them fully, but it's only an incomplete enveloping of the capillaries. So, okay. Now you can see the blood-brain barrier. I think you can just choose one figure to draw. This blood-brain barrier normally comes as a 5 marker or a 3 marker for you all. Now let's look at the movement of substances across the blood-brain barrier. Now the movement of, see what substance are going to go into the blood-brain, cross the blood-brain barrier to and fro, it depends upon certain properties of the substance. For example, the size of the molecule, the lipid solubility, the ability of the substance to bind with protein, the pH of the body fluids. So these 4 factors determines the ability of a substance to cross the blood-brain barrier, either get into the brain or come out of the brain circulation. Now these substances can move mainly by 2 or 3 ways. The vesicular transport, the carrier mediated transport, and there's one more, not a major one, which you call as the facilitated diffusion. I think I have mentioned only two. Yeah, but I think you can always add the third one, the substance, by the two ways. The most important one is the vesicular transport, then you have the carrier mediated transport, and the last one is the facilitated diffusion. Now a little more into the basically the permeability of substances. You can see that carbon dioxide, water and oxygen passes through the blood-brain barrier very easily as they are neutral substances, isn't it? So neutral substances like water, oxygen and carbon dioxide can pass through any membrane. So same way, they pass through the blood-brain barrier because the cells needs to be oxygenated, isn't it? It's doing a very major function. Now lipid soluble substances like alcohol, anesthetic agents and steroid hormones also can pass easily. H plus ions and bicarbonate ions penetrate, but they penetrate or cross through the blood-brain barrier very slowly. Now the fourth, we have already mentioned about the lipid soluble substances which can readily pass through very easily. Now what about the non-lipid soluble substances? Now the non-lipid soluble substances cannot pass through the blood-brain barrier. Now several drugs, like, so you have a look at those ions there, Na plus, K plus, Mg2 plus calcium, bicarbonate ions, the phosphate ions, all of them can pass, but they pass very slowly. If you look at glucose, the glucose transporters are there, and the amino acids, they are getting transported by facilitated diffusion, which I mentioned as the third point, the mode of transport. Now the bile salts and the catecholamines are not able to enter the adult brain. Now several drugs like dopamine and bile pigments also cannot cross the blood-brain barrier. So here you have, okay. Now I think on the permeability of substances a little more in detail. If you go, when you specifically talk about the drugs which can cross the barrier, sulfonamides and erythromycin enters easily, but penicillin and chloromycetin have very limited entry. Antidepressants and some psychotropic drugs which are fat soluble can enter easily. If they are not, the antidepressant and the psychotropic drugs which are not fat soluble cannot cross the blood-brain barrier. Anesthetic and thiobarbitone can enter easily, but barbitone does not. Dopamine and serotonin have very limited entry, but L-dopa and 5-hydroxytryptophan can enter with very high pace. Now let's have a look at the development of blood-brain barrier. When exactly does the blood-brain barrier develop? At birth, the blood-brain barrier is never fully matured. Now the functional maturation of the blood-brain barrier will be completed by 1 to 2 years after birth. One second, let me just check out where the functions of, I think I need to do that. Okay, now if you look at the functions, now you know how the blood-brain barrier is formed, what is the blood-brain barrier or the blood-brain barrier, CSF barrier, all that, what it is, how it is, what are the substances which can pass through the barrier, all this you know. But there's one important thing, I don't think I have mentioned in the PPTs in this, I've just missed it, but you can just keep a note of it. It's very simple. 3 important functions of the blood-brain barrier. Now the blood-brain barrier, it maintains a constant chemical environment around the neurons, which is very much essential for their proper functioning, and any fluctuations in the concentration of K plus ions, the calcium ions, magnesium and the H plus ion concentration can always alter the neuronal activity. Number 2, it protects the brain from endogenous and exogenous toxins in the blood. The third one, this also prevents the escape of neurotransmitters which are present in the brain into the general circulation. So these are the 3 most important functions of blood-brain barrier, where it maintains a perfect chemical environment for the brain to the neurons to function properly, because any fluctuations in the concentration of potassium, calcium, magnesium ions, etc. will alter the activity of the neurons. Point number 2 was, it protects the brain from both endogenous and exogenous toxins, and one more thing is, it prevents the neurotransmitters of the brains to escape into the general circulation. So now we go to the applied aspects. The applied aspect is something like, the physician should have, the treating physician should have a clear knowledge about the drugs which can pass the blood-brain barrier while treating a patient. For example, penicillin and chlorotetracycline, it enters the brain very slowly and to a small extent, whereas sulfadiazine and erythromycin can enter the brain very easily. Similar, dopamine cannot pass through, but L-dopa can enter the brain freely. So this knowledge should be very clear-cut for the physician. Now, if you look at jaundice, now the jaundice in a newborn which can cause a damage to the basal ganglia because it gets deposited in the brain cells where you call it as the kernicterus. So what happens is in newborn, the blood-brain barrier is not very well developed. So the bilirubin enters the brain freely and this bilirubin deposits in the basal ganglia and it will cause a damage to the basal ganglia. So now the third thing is the blood-brain barrier can get disrupted in a person under following clinical conditions. For example, a bacterial meningitis, ischemia, brain tumors, brain injury, radiation, all this can cause the damage to a blood-brain barrier. Now congenital disorders where there is deficiency of GLUT1, when there is a deficiency of congenital deficiency of GLUT1 in a person, that will lead to a decreased glucose level in the cerebrospinal fluid, as this GLUT1 is responsible for the glucose transport into the brain. So when GLUT1 receptor itself is transporter itself is deficient, means naturally the glucose levels in the CSF would drastically go down. Now children with this deficiency, there is a congenital deficiency of GLUT1 transporter, they will develop convulsions due to decreased glucose levels in the cerebrospinal fluid. Now a little more on the blood CSF barrier. If I tell you, the capillaries of the choroid plexus, I think, okay, there's no picture there. The capillaries of the choroid plexus actually show gaps between the endothelial cells of the capillary, but the choroid epithelial cells that separate them from the CSF are joined by very tight junctions. So I just repeat that. Now the capillaries which are present in the choroid plexus, actually between the endothelial cells you can see gaps. But the choroid epithelial cells that separate them from the CSF, you can see they are very tightly joined by tight junctions. Now, however, there is no CSF brain barrier as the drugs injected into the CSF by lumbar puncture reach the brain and the spinal cord. That's a very important point to be noted there. There is no CSF brain barrier when the drugs are injected to the cerebrospinal fluid by a lumbar puncture, which can reach the brain and the spinal cord very easily. Now let me go to the small part of the blood-brain barrier where you talk about the circumventricular organs. Now what do you mean by circumventricular organs? Now the circumventricular organs are structures of the brain which do not have a blood-brain barrier. Now these are located around the third ventricle. The capillaries, if you see, are highly fenestrated and they are highly permeable of these circumventricular organs. Now this essential is to maintain their physiological role, or else in simpler words you put it as this, this highly fenestrated, highly permeable arteries helps to maintain homeostasis. Now to control blood pressure, fluid and also electrolyte balance. Now exactly for you all to know which are the 5 circumventricular organs present in our body. You have the posterior pituitary, subphonical organ, organum vasculosum of lamina terminalis, area postrema and median eminence. Now they are present outside the blood-brain barrier so that they can be influenced by the blood-borne factors. So you have the posterior pituitary. So what happens here is the lack of, so you can see them in picture, all those structures which are, now the posterior pituitary, if you look at, one second, yeah. So the posterior pituitary, if you look at this lack of blood-brain barrier here at the posterior pituitary, it facilitates the entry of posterior pituitary hormones into the bloodstream. See, that's exactly why the reason there are few organs which are categorized under circumventricular organs which doesn't have a or which is out of the blood-brain barrier. Because now when you look at the posterior pituitary, why is posterior pituitary out of the blood-brain barrier? The reason is only then the hormone secreted by the posterior pituitary can enter the bloodstream. Now, the median eminence of the hypothalamus and the pituitary gland, it enables the hypothalamic hormones formed in the median eminence and the oxytocin or the vasopressin to enter the blood of the posterior pituitary. So I just made it more specific. Previously when I just grossly mentioned it as hormones. Now the area postrema, it contains a chemoreceptor trigger zone which initiates vomiting. It is also involved in cardiovascular control. The third one, the organum vasculosum of lamina terminalis ovalt. They actually may have osmoreceptors which control the release of vasopressin. The fourth one, you have the subphonical organ or the SFO. Angiotensin II is believed to act here, and it increases the thirst and the water intake. So I think even pineal gland, I've just missed to mention that. Even pineal gland, there is a lack of blood-brain barrier, which actually helps the gland to release melatonin into the general circulation. So So basically all these structures, these 5 or 6 of them, they are located around the third ventricle, and hence we give the name circumventricular organ. I think you can see that in the picture. You can see them all surrounding the third ventricle, and these are called as the windows of the brain. Now the capillaries in this organs are highly fenestrated and are highly permeable. So hence they are said to be present outside the blood-brain barrier. Now this particular arrangement around the third ventricle, which we call as the circumventricular organs, is highly essential to maintain their physiological role of homeostasis. So I think that's all about the blood-brain barrier and the blood-CSF barrier. Please be thorough with that. Either a 5 marker or a 3 marker can come from the class. I think this is your last theory class, I believe. So we'll be continuing with the practicals and also the revision classes. So study well. So thank you guys. Thanks for listening.